I wanted to put together just a quick video with some guidelines and some helps for lab number 2. When we're doing Evo-Devo labs, there are a number of different aspects of a project that need to be addressed, and some of these are very labor intensive. First, you need to get familiar with the organism that you're working with. In the past, we've done a lab that's based on Drosophila, a model organism. Now, the nice thing about that is that a lot of work's been done. It's easy to do because all of the methods are pretty standard and have been worked out really, really well. The pandemic has made a hands-on wet lab really not an option for this semester, particularly for the size of this course. So the modifications I'm making are that we're going to do an analytical lab. Basically, we're going to analyze transcriptomes, look for genes, and we can do that remotely. But that doesn't mean that we don't need to be familiar with our organisms. Now, the downside of working with Drosophila is that it is work that's already been done. So you're really not doing anything new, which is fine for most teaching labs, and that's okay. But for this lab, I thought maybe since we're going to do it online also, and we're going to analyze data, we might as well analyze new data, because the analysis of this data is really the same no matter what you're doing. And there's not really that much new. There are a little bit slightly different approaches if it's an unknown transcriptome versus from a known one, but nothing huge and that we can't overcome. So with that, I want you to get a little bit familiar with the organisms that we're going to be working with. We won't get data till about lab 4, maybe lab 5, depending on how we're able to generate that data. It's in the works right now. But I want you to start getting familiar with the organisms. So that's the purpose of lab number 2. We're going to be working with different insect transcriptomes, and each student will be assigned a different insect group. And then as transcriptomes become available, I may make some slight adjustments and move you to a different group for which a transcriptome is available. Now, the second part of any project is looking at the actual, we're going to be looking at genes. So looking at the genes, the genetic system, the history of those genes. We will be doing that in lab 3. So lab 3 will be kind of our last foundational one, where we're going to be looking at a history of the genes and some of the information about the genes that we will be working with. Now, for this lab, we're not going to do the classic Evo-Devo genes. And the reason for that is because in an Evo-Devo project, the third major group of skills that you need to pick up is the embryos, the working with, keeping animals so you can get embryos, or else getting out into the wild and gathering enough embryos and preserving them and fixing them so that you can then work with those embryos. And that does require a lot of hands-on work in the lab, and is really not feasible the way this lab has set up this semester. So the downside of that is we're not going to be doing kind of a classic Evo-Devo project. However, the methods we're using, the transcriptome analysis, transcriptome assembly, gene identification, can all be applied to Evo-Devo stuff. And in fact, I will have you do and locate a few Evo-Devo genes, even though they're not going to be the primary target, and we may not even expect them in the adult tissues from which our transcriptomes are derived. Okay, so that's just a little bit of information, just so you know what to expect. If you have any questions on that, if you want to go into that further with me, you can make an appointment with me during office hours. Now, as part of what we're doing for today's lab, I'm asking you to do a very short literature review on an organism to which you have been assigned. And all those instructions are in the lab itself. So when you get to that part, it's near the end of the lab, you can use this section of the guide video to help you. I'm going to suggest 2 resources that you can use to complete that literature review. Now, you do not have to use these resources. If you have others that you are familiar with, or if you want to just do it the old-fashioned way for the literature cited section, that's fine, but these will make your life easier. In the short run, there's a little bit of a learning curve, but in the long run, especially if you ever have to do another paper with a literature cited, which all of you will at some point, maybe multiple times in your college career, this will really make generating the literature cited and getting that all in perfect form a lot easier. Okay, so the first resource that I recommend you use, again, if you've got others that you think will be sufficient, that's fine. But the first one is a database called Web of Science. This is a subscription database. However, the university subscribes to it, so all students and faculty have access to it. Now, if you're not on campus, you can't directly access it because they have a gateway that recognizes who's subscribed and who's not. However, to access it as a student, you go to the UTRGV library page, and then you click this little link here, which will allow you to log in. You'll have to put in your university username and password, and then you will be logged in. Once you're logged in, then there's a link here on the main page that is Find. It's under Find Databases, and we're going to go down to W for Web of Science. This is a fairly extensive, I've got to put my password in again. This is a fairly extensive database that does a really good job. No database is going to have every single literature, especially if you're getting to very, very old scientific literature, but for our purposes, this will be more than sufficient. And there are other resources. You can use Google Scholar. You can just Google it sometimes. The second tool that I'm going to show you, which is a citation program that's freely available, also has a search that's pretty good also, but not quite as complete as Web of Science. So I'm probably going to suggest that you use Web of Science if you don't have one that you feel is really good. And even if you do, you may want to use this one and see if it's better or get used to it. So I'm here at Web of Science. I'll come back to that and demonstrate this in a little bit. Okay. The second tool that I want you to investigate, again, if you feel comfortable or you don't want to bother, that's okay, but I really highly recommend it. And it's this program called Mendeley. Mendeley is a literature citation management software program. Big, long name, but you can go to mendeley.com, E-M-E-N-D-D-E-L-E-Y, okay? And you can download it for free. There's a free version that's really a very functional version. Now, not only will it keep track and store all of your references for, not just this paper, but any other research you do, and you'll always have those there. It will also give you a tool that will create and format your literature cited section. So you don't even have to do that. And if you've ever written a scientific paper that required a literature cited section, that can be a huge pain at the end to make sure everything's in the right format, type it all out, add, and then if you need to add or subtract new ones. And it's even worse if you need to change the format, which is rare for classes, but happens sometimes if you decide you want to submit a manuscript to a different journal and you need to change the format for it. But Mendeley will do all of that for you for free. So you can download this. There's a plugin for Microsoft Word, which I've downloaded and I'll show you, and works really, really great. So I'm going to demonstrate how to do this and give you a little bit of guideline of how to complete this literature cited section. So I have provided a template for you for the literature cited. So I'm going to open that up here. And I've just called it literature review template. It's a Microsoft Word document. It's very simple manuscript format, very easy. So you'll put your name here. You're going to put your assigned group, which is found in the document here in the lab folder. So groupassigned.pdf. Look that up. Open it up. Find your name, so whatever your name is, and you have your assigned group. I've also given you the common name. Some of the common names are just like the scientific names, but you're probably going to want to use the scientific name when you're doing searches. So whatever group your name is assigned to, that's your assigned group for this project. And I may make some adjustments depending on transcriptome availability once we get to actually analyzing the data in later labs. But for now, that's your group, and that's what you're assigned to do a literature review on. So I would put it here. Now, just to demonstrate, I'm going to pick one that's not part of the group so I don't do the work for anyone. Everyone needs to do their own work. But let's say you'd been assigned the group Scorpions, which their scientific name is Scorpiones, and that's what you're going to do. So put your name, your assigned group here, and then we're going to replace here, I've just put body of literature review. We're going to replace that. And so you're going to do a little bit of work on your assigned group, and then you would say, the group, you can just, you don't need to write this exactly. I'm just going to start. The group Scorpiones is composed of, I don't know, and I haven't done the research of this, but I'll begin that and show you at least how to begin, of 4 major subgroups. I'm making this up, so I would need to double check. But you're going to give me a short, it just needs to be a page to a page and a half, 2 pages max. But you're going to give me a very short summary of what is known about the group, including literature cited. So to do this, once you've got your group assigned, you would go to your choice of database. And I'm going to open up the Web of Science, which I've gotten here. And so I'm going to search, just keyword search or topic of the paper, for scorpion phylogeny. Now, this database, many do this, by default arranges them in the most recent. So whatever has been published most recently will be first. Now, with a general term like that, I'm going to get a pretty broad result, 252 results. Many of these are going to be not really exactly what I want. They might be like a subgroup within the scorpions and looking at the phylogeny of that. It might be a paper that's primarily about another topic in scorpions but mentions phylogeny or refers to phylogeny. So we want to look for papers that are specifically about phylogeny of the group. And so maybe if like, well, okay, that's a little general. Maybe if I put the scientific name here, that will be a little bit better. So Scorpiones, phylogeny, search for that. And see, I've narrowed it down a bit. I've got rid of about half of the ones there. And so I'm looking for titles now. Genetic diversity among different species of a genus within the scorpions. That's not really what I want to do among populations of this one species. So I'm going to search down genetic diversity within, again, a single species, interspecific phylogenetic relationship among different species of, I was getting excited because it looked good, but now, again, it's just one of the genera, so one genus within the group, but it's really not what I'm looking at. A Cambrian Ordovician terrestrialization, that's maybe talking about fossils, probably not what I want. Developmental gene expression, now that's kind of cool, support for monophyly, but not maybe. I want to look at the ones that are primarily. Major revisions in arthropod phylogeny. So here's one that might talk about scorpions, but it looks like it's maybe a higher level of the big group. So, and notice we're still in 2020, so we haven't gone back that far. So you might need to do a little bit of work here, and you may even want to use a combination of resources. So I have also, and this may be different for different groups, I've also put the exact same search term in the literature search section of this Mendeley app, which is a little general. So we might miss some papers, but because it's general, you're probably not going to get as a massive amount. So notice it just kind of gives me an overview. And the very first one looks really promising. Phylogenomic resolution of scorpions. So looks at like the whole thing. So I can click on this and it'll give me an abstract. Scorpions represent an iconic lineage of arthropods. Oh, look, higher level. Oh, this is exactly what I want. And it's recent. It's only like 5 years old. So this would be like maybe the key paper that I would like to use to give me an overview and a summary. So I need to, if I'm doing a literature review, which is your assignment for this, I need to find this paper. Now, this is the nice thing about Mendeley. Once you've found it here, once you've searched for it either by title or general topic, you can save that reference. Now that reference is part of my library, and I'll show you what that means here in a little bit. Okay, but in addition, I would like to find this paper. Now, I don't have a link here to a PDF or an online version of this paper that I can read, so maybe what I can do is, well, let's copy and there might be other ways to do this. If you find other ways, that's great. I'm going to come back to Web of Science, and that paper is almost certainly on this list. So I could either say, well, that was published in 2015, so I could narrow my search, search within results for 2015. And probably find it that way. All the papers about scorpion phylogeny published in 2015, there are only 5 of them. And funnily, I don't see it here. That's funny. Maybe it's looking for 2015 as a key result or a keyword instead of the year it was cited, because, yeah, these are from a different 2016, 2015. So I might be doing something funny here. So instead, I'm going to go back to my original main search thing, and I'm going to enter this and say, let's look for the title. And there it is. All right, so we can find it. Oh, look, free full text from the publisher. Great. That's what I want. Here's a PDF of my recent paper about scorpion phylogenies. Now, if you find the most recent paper, and for some of you, there are going to be dozens and dozens of paper in your group, so you maybe only need to look at the 2 or 3 most recent ones. Whereas for others, it might be a little skimpy, and you may even have to kind of stretch and look and maybe include a paper that is a phylogeny of a bigger group that includes your small group. But for scorpions, there's a lot of work done. This one's going to be very recent. And one of the cool things you'll almost always have in a paper that's this kind of scope is a summary of previous work. So they're summarizing 1, 2, 3, 4, 5 previous papers that looked at scorpion phylogeny. So they've already done pretty much all of your literature review for you and even summarized it. So you can refer to these papers and say, maybe do a quick summary based on what they've said here about, okay, this person said this and these people said this and they agreed in this way, but there's disagreement, and our most recent one, and there's going to be a diagram here showing the most recent phylogeny of major groups. I don't know if that's the best one, maybe farther down. Oh, here we go. This study, here's their summary of the major groups within scorpions, with some look like they're paraphyletic. And so you need to do enough of a work, and this will stretch you a little bit, I understand that. You need to do enough work to be able to give me a very short summary of the phylogenetic relationships within your assigned group. And again, it's short, a page, 2 pages and a half max. So that means it really is a brief summary of our current up-to-date understanding. But you need to get into the literature and do that. Now, once you have a paper, and you can use your other app if you want or do it by hand, but once you've got a paper in Mendeley, there's this really great option where you can add a citation. So let me show you how to do that. So here's my word template. The scorpions, again, I need to read it to do it. But let's say I wanted to reference this. I'm saying something, I'm making a specific claim here. The group Scorpiones is composed of 4 major subgroups, and I want to add a reference because maybe that's what this Prashant paper, the Sharma paper said, and so, okay, I want to reference that Sharma paper. So I can do this because I've put the Mendeley add-on here in Microsoft Word. And so I can now insert a citation. It's a nice little thing there. I want to say, I want the scorpion. I don't remember exactly what it was called, so let's search for it. Oh, there it is. There's the paper I want to add. I'm going to push OK. And notice that it adds it automatically. It puts it in here. Now, not only did it put in the text, it also put in this little a widget. I don't know what it would be called specifically, but this little tool that keeps track that is a citation. It's a little different than just regular text. And so at the end, after I've inserted all of my citations, I can say, oh, I want to insert a bibliography. And it will automatically, let's get it to the right place. I want to do it down here underneath here. I want to insert my bibliography. It will automatically insert a bibliography with the correct format. And I say, oh, wait, Dr. Terry has asked me to do a different format. And so let's pick a format right now. Let's do, let's see if we can do this, molecular phylogenetics and evolution. That's a good format. It's a pretty basic one. Many, many journals use the format they use, but this is the journal the format that journal provides. So I'm going to say use this style and say done, and it will automatically change the format for that citation. Okay. So again, use Mendeley. A little bit of a learning curve maybe, but it will greatly do it. So let's say, and I've got a bunch in here from other previous papers. I like Mendeley and it works well. Let's say I wanted to insert another one. So I want to insert one that's just about phylogeny in general. So and I do a lot of stuff on phylogeny, so I'm going to do that. And so now notice it automatically, I didn't even have to do anything. It automatically inserted the citation for me. So it's wonderful. It's really, really great. Does a really good job of making your life a little bit easier. Oh, look, there's another one. This is funny. This is from 2005. I did actually have a scorpion phylogeny paper. I just noticed that I had previously downloaded. Prindini and Wheeler from 2005. So it's a little bit older, but there we go. Oh, no, that was in my search. So anyway, save. Oh, let's say I want to add that. I like to add that one too. Now, Also, notice I put spaces here, but if these are all in the same ones, if I add them right next to each other, I'm going to have a little bit, it should put them all together. Let's do it. Let's add a bunch of them. So let's say I wanted to recite 3 of them here in the same place. So I'm going to go in and I'm going to say insert citation. I want the scorpion one. I want that one. I also want this other scorpion one that's more recent. There's that one. And then I'd also like to put in maybe this ostracod one, which really wouldn't go there. But so anyway, I'm going to say, okay, there they all are. It's going to put all 3 in the right place and going to put all of them down in my literature cited. Okay, hopefully that's helpful. Once you are done, again, just a page to a page and a half summary of major phylogenetic relationships within the group that which you've been assigned. Save it as a Word document. I would just go ahead and save it. You can give it any name you want. Attach this and your completed lab to the lab assignment. And now remember that the due date on the calendar is Friday, but I give you until Saturday, so I put the deadline until Saturday. You can turn it in Saturday if you'd like. Better to do it by Friday. As always, if you have any questions, if you get stuck, send me an email, log on during Zoom office hours. I'm happy to go over with you, even answer questions. I probably will try to talk you through them rather than just giving you the answers. But I'm here as a resource for you to use if you get stuck or having trouble with any of the parts of the labs.